So welcome everybody, and thank you for taking the time to come here and listen to the results of the FlyScent project that we have been running over the past few years. I will go through the broad strokes of the project, what was the aims and what was the work that we did, and what was the focus on the different parts of the project. That, as well as I will also present the partners that were in the project and their roles in the project. So we're talking about FlyScent today, and the subtitle for the project was Formulated Fly Perfume for Consistent and Directed Oviposition. It's a Eurostars supported project with a total budget of around 1 million euros. This is me. I am a specialist at the Danish Technological Institute. I started here 6 years ago. I started as an insect expert. My PhD was on cold tolerance of insects, trying to investigate what happens to insects when they're exposed to cold. And that is actually part of what we have done also in the recipe project that many of you may have heard of. Over the past couple of years, I've been moving more and more towards becoming a data analysis expert, actually. So I'm leaving the insect work mostly to my colleagues, but then I'll help them when it comes to analyzing the data. This face, I'm sure many of you know. This is Anton. Anton was the one who actually came up with the project with FreezeM and wrote the application and won the application, and he was running the project for the first half period. Then he left us to pursue the title of Dr. Glikorescu. He's now doing a PhD at Aarhus University, which is something that he wanted for a long time. So I guess we'll have to forgive him for leaving us. This face, some of you surely will know. This is Jens Fynbo. Jens is one of the two partners in the project, and unfortunately Jens cannot attend today. I talked to him yesterday. Jens has a concussion and has been unable to prepare and unable to sit in front of a computer the whole week. So I promised Jens that I would put up his contact information on the screen. So if you have any questions regarding the technologies and the equipment or something specific related to the insect industry in regards to machinery, Jens is the one to go to. So you can send him an email now, and if you're going to call him, maybe give him a week to recover from the concussion. All right, that means that there is a slight change in the program. So this is the original program that was uploaded. And what we're doing is that my colleague Nida here is going to present Jens's part. So Nida's talk will be extended to half an hour, and we'll be talking about both egg collectors, distribution system, and experimental results. Luckily, these two are very intertwined because it's the egg collectors and the distribution systems that were tested in the experiments that Nida is going to talk about. So we postponed the break a little bit, and then we'll catch up after the break and be back on track for the remainder. All right, an overview of FlyScent. The main aim was to make an integrated system that has an attractant that can guide the female flies to lay eggs where we want them to lay eggs and not anywhere else. And that also included figuring out a method of dispersing this scent that is supposed to lure the flies. I'll talk a little bit about the consortium and then the work that we did. So yeah, the aim of FlyScent was to develop an effective, easily implementable and cost-effective oviposition solution for the reproduction of black soldier fly, because we know that the reproduction is actually a little bit harder than it looks on paper, and by far the hardest part of the black soldier fly production, or the most risky part at least. So the aim was to make an attractant that would attract the flies, and this was done through a series of experiments with biological and chemical compounds that were characterized, tested, and their ability to attract the female black soldier flies to control the oviposition. Quinn will talk a little bit more about specifically the screening of the compounds and the isolations and what was done in that regard. Then there was the dispersion solution, a scent dispersion system and egg collectors that fitted the system that was developed and optimized during the project. Jens was supposed to talk about half of this, but now Neda is going to talk about all of it. And finally, there were large-scale tests of the products that we developed. So the large-scale test was done in FreezeM's production setup, and then we had smaller, more controlled setups here at the Danish Technological Institute. Neda will talk about the small experiments, and then Yuval from FreezeM will talk about the large-scale experiments. So the attractant work included, among other things, these points that I put up here: literature review, chemical characterization. There was a safety and regulatory assessment. There was different dispersion methods to be tested, and easily implementable egg collectors, and finally an integrated oviposition solution that were ready for market. Testing and optimization at lab scale, we looked at different oviposition attractants, we looked at different dispersion methods, we looked at different egg collectors, and again, we looked at the best solution for an integrated system. Pretty much the same with the demonstration, just on a larger scale. Demonstration of the integrated solution, identification of possible bottlenecks or pros and cons, assessing the suitability for the entire industry, because all of you have very different setups, and we were trying to figure out how to make this work for the most factories possible. And finally, there's a business assessment looking at what this could actually save in regards to money and work hours. Finally, there was a part in the project, of course, there was dissemination. That's also why we are sitting here today in this workshop or webinar. There is going to be a small report on performance of some of the experiments that we conducted. And finally, there will be a peer-reviewed paper with some of the results from the project. And that is pretty much all I have to say for now. I'll put up Jens's information again. Again, Jens is a technical expert. He can build you robots or pallet movers or whatever you need. And if you have any questions in regards to these things that we did in the project, you can contact Jens. All right, that's all I have to say.
Perfect.
Back to you, Mads.
Thank you so much, and thank you for that presentation. I can see for the moment there's not any questions, and that normally means you did a fine job. So I'll use that as a testimony.
Anything.
I as well have a little bit of question, maybe a weird one, but have seen FlyScent from the outside, and then now being on the inside, I was wondering, do you have the same experience that projects like this, like the FlyScent, they change a lot over the time or the time period?
You mean the project itself changes?
Yes, so like the scope of it.
Yes.
Did you have the same experience with this?
Actually, this one didn't change that much. The ideas that we had in the beginning of how to do this changed a lot, because we could see that some things work and some things don't work. But the general idea about making an oviposition solution that everyone can implement fairly easily, I think we actually reached that point, or are going to reach that point in the final 14 days of the project.
Amazing. Awesome. Thank you so much, Jonas, and I will be moving on. And in advance, I have apologized for the pronunciation, but the next presenter is going to be Quinn Nguyen, something like that. At least she works at the Center of Air and Sensor Technology here at DTI. She's a chemist with the PhD in atmospheric chemistry. She works a lot with scents, is probably the best way for me to say it, and as well volatile compounds, and have been doing some really interesting parts of this project. So can please take the floor.
Screen got froze.
Yep.
Yes, awesome. Yeah, so I will be following up this dissemination on the results from the FlyScent projects by talking about a development of chemical attractants and a.
I see your presentation. There's my computer. There's my computer.
Yes, sorry that there was some the technology that freezes. Anyway, a chemical attractants, and we need chemical attractants because of the positions of black shoulder flies today. It's
just a moment. We don't have
Okay.
it.
Yeah, that doesn't work. You have to use your own computer. We can change the
I
slides,
can
but we are
share.
not changing them for
I
everyone.
share Queen's presentation.
sorry everyone. Apparently there was a glitch somewhere, and mine is not shown.
That's fine.
I hope it's fine now. It's fine.
1 minute. Wait, 1 minute.
Nina is sharing your presentation now, so you can use her computer.
Okay.
It's probably fine.
I hope you see a right screen that's shown development of chemical attractants.
Yes.
Yep. Okay, that's good. Right, and we need chemical attractants because the situation for black shoulder flies of the position today is that we're basing a lot on this decaying organic materials as attractants, and these are quite difficult to control. And this might lead to high egg losses due to oviposition across the entire cage. On top of that, there might be risk of mold and bacterial contamination, as well as egg infertility due to pathogen. And so we were like hoping for in this project to develop chemical attractants that are reproducible, controllable, scalable, and bacteria and pathogen free. Yeah, well, like hearing all of this, then it might be like, okay, how do we do this? And this is something that could be something like too good to be true in that direction, or if it even works, or and how do you describe a chemical smell, and do the flies like them or not? So we'll start by introducing a little bit on chemical signatures of smells. Smells are characterized by volatile organic compounds, and there can be complex smells or singular smells. For example, the singular smells could be when you have an orange or lime. That smell is a limonene chemical. Or if you have a banana, is isoamine acetate. Or some bad food has some signature smell as well. Or if you step into like a pine forest, then that pine fresh air that you breathe in is pinene. So all these, they have different signatures. Now, when we think about the smells to the flies and how do they interpret it, then it becomes interesting because we don't really know they smell the same way as we do, or how do they experience it? Do they like, as in like bursting smell with a lot of signals, or do they prefer simple smells, or what kind of like, how strong should the signals be? Yeah, there's a lot of questions, and we can speculate that the smell of the volatile organic compounds here, they interpret it as information on a source of food, some sort of like nutrients that they could get from the environment. Yes, so to develop our chemical attractant, so we have taken the approach by first looking at a chemical screening of potential compounds, and that was done by combining literature review and investigating the chemical contents of potential biological attractants that work. And then in order to characterize these chemical contents, then we use a technique called PTIMS, which is short for proton transfer reaction mass spectrometry, which is a mass spectrometer and quite a state-of-the-art instrument, which allows you to measure volatile organic compounds of quite broad spectrum, up to a mass. Yes, a bit specific, but up to 300 mass as in molecular weight. And you can use this instrument to obtain chemical fingerprints of biological attractants, and also follow with these fingerprints signals over time. So it's how we characterize it. Now, so back to building blocks. We started with to build up this FlyScent, or the flight chemical attractant, by looking at literature review and look at a lot of things like what are the volatile organic compound signals during food decaying process. We also look at like the death signals, for the decaying, decomposing signal. That was actually from a forensic journal. So it's like, it's actually quite broad, the materials that we look at. We also look at the different types of feed that could be attractive for the flies, from for example, chicken feed to fish feed, and different types of literature. And as mentioned, we look at potential materials, as in we have our own DTI biological attractants. We also look at different amino acids, fermented carbohydrates, and we look at the chemical fingerprints of these compounds. And this is actually a picture of the PTMS instrument in action, where you have this, where you have the compound here, or the instrument here in the corner, and measuring the signals from the different containers containing the attractants, the biological attractants. Yes, and from that, then we built sort of like a small database of the signals and concentrations, based on a lot of signals from this PTMS instrument and how they evolve over time. Then we try to identify the odour compounds, the potential ones. We look at their presence, their concentrations, and one very important thing is the odour threshold values, because some compound can be presented in like small concentrations, but then they are like really smelly. So it's also one of the indicators that we use to shortlist the compounds. And as a result of this shortlisting, then we identify 20 different chemical compounds. Now, I can't really share this list, but it is like this sensory description of these 20 chemical compounds that we shortlisted. And you can see that it's actually quite broad. Then it ranges from fruities, banana, cantaloupe, to like rotting egg, rotting fish, or to Asian market smell. So it's actually quite broad description that I can get here. Now, when we shortlist it, then we need to find a way to generate the smell based on the chemical compounds. Of course, we purchase the shortlisted compounds home, and the idea of generating the attractant or the FlyScent here is to find a way to disperse this chemical compound or solution. But then how is a quite difficult question, and it requires a lot of experiments. But then here is an example where we show some of those the vials containing different matrix of the potential compounds, and where we put them into some sort of like gray cube here on the table, and we feed this into the PTMS using a pump system. And we follow with the signals and evaluate the stability of the signals. And in the end, and the compounds of interest, then we could achieve quite good stability in the good concentration range of up to 2 days. Yes, this is one example of how to generate a FlyScent in a more stable way. So we use some water beads to, so that is like some materials that we use a lot for decoration or in flower settings. So it's some sort of materials that it sucks up the water, and also you can use the different chemical solutions for it to suck up. And once it's absorbed all these solutions, then it will release this scent like slowly over time, and with that then you can kind of like slow down the process and preserve it more. And once we get all these cubes or different candies of different colorfuls, colors, or different solutions, then we use then we place them in different experiments. When it is one setting with the black soda flies in lab, where we put them into different gray cubes again and then feed them to different. There's some of those egg collectors on top, which my colleague is going to talk about more later. But by that, then we could feed the obtained chemical solutions to the flies in a controlled way. Yes, as a sum up for this part on development of chemical attractant, then we have found several good potential chemical attractants, which we could generate the concentrations in a controlled way and stay quite stable over up to 2 days. And we found one winner. It's a compound X. But my colleague will be talking a bit more about like how the experiment of how to test this in a lab testing together with the egg collectors. Thank you for your attention.
Thank you so much, Quinn, for that presentation. Really interesting. Same with the Jonas goals. It seems like you presented everything just to A+. Although I have a small question I'd like to ask. So, from your opinion, do you think like chemical smells or biological smells is the, let's say, the answer for attracting black soda fly ovipositioning?
Yeah, I think it's in this one. Then we're actually trying to look at another approach. So basically, it has been a lot of biological smells before, and chemical smells is kind of like the new way. As I mentioned, there are like a lot of issues that we could see with biological smells. So we were looking at a way to do it in, with the chemical fingerprints, and according to this, and there's actually quite good potential, and the flies seem to like them. So I was quite happy about it.
Yeah. Also important that the flies like it, definitely. So I think we will we'll go to the next presenter. It was a hand, I'm told.
Yeah, come on.
Yes. So there's a question. How much are these potential compounds related to the food that the black soda fly ate in their lifetime?
I think
that's a perfect question.
We will come with the answer of this question in my presentation, if you just stay a little patient. Thank you so much.
That is a very intelligent question.
Yeah, that's a really good question.
So from that, I can hear we will come straight back to that with Neda, I think, with presenting this. So without further ado, I'll continue with introducing Neda. She has been working several years with different insects. Right now, she's our breeding expert on black soda flies, and she'll be presenting the ovipositioning solution we have designed for this project. And I will, with that, leave the floor to you.
Thank you, Mads, and thanks everybody for joining this webinar. In my presentation, I will share the results of experiments we performed during the FlyScent project to develop an integrated oviposition solution for black soldier fly. I have divided my presentation in 2 parts because it's a bit long presentation. Hope I don't make you confused. The first part of my presentation is about the journey to develop another attractant, and the second part is about the egg collectors that we developed and we looked at different factors of egg collectors regarding color, shape, materials, and also we developed a feature that enables insect producers to collect and separate eggs from the egg collectors without scrubbing and without damaging eggs. But why do we need an odour? In natural conditions, black soldier fly females usually lay their eggs close to a food source to be sure that when the small larvae neonates hatch, they will have immediate and easy access to nutrients. So to target black soldier fly females to an oviposition sites, usually insect producers use decaying materials, fermented fruits or frass to attract females, and they use these materials as bioattractants. But all these attractants have one common issue, is that they may be contaminated with pathogens that increases the risk of disease outbreak in populations, and it may also affect the fertility of females. Plus, preparation of these odours is a bit labor demanding, and these odours are not able to produce a consistent odour during their production. So all these factors together has influence on the reproduction stage of black soldier fly, which is a very important part of the production of these insects at the industrial level. To cope with this issue, one of the main aims of the FlyScent project was developing an efficient, safe, and consistent odour for black soldier fly. To reach this aim, as my colleague Queen explained, we started by a literature review. We came up with different candidates. One was an amino acid, another one a fermented carbohydrate, and a commercial odour. And we assessed the efficiency of these attractants to attract black soldier fly females towards an oviposition site. And we used our DCI attractant as the control ones. At the same time, we also looked at the components, the volatile components that release from these attractants to find a correlation between those components and oviposition behavior of flies. The graph that you see here shows the amount of eggs we collected from the different places that we had the different odours. And the DTI was the winner, but we were so happy that flies they showed response to other attractants, like the amino acids, fermented carbohydrates, and also the commercial odour. The results of the PTRMS gave us many components. It was so complicated, so to make this complicated pattern simple, we used a specific statistical analysis known as PCA to find a pattern between all these arrows that each of them shows one or belongs to one component. To make it more understandable, in this analysis, usually based on the presence of the arrows, regards to the arrow of the egg production that is shown in green color, we understand that which component has a significant influence on the egg production or the attractants of flies. Based on these results, we selected some of the components and we did a new test where we assessed the influence of one component, mix of 3 components, and mix of 8 components on the oviposition behavior of females, black soldier fly females. We use again DTI as our control odour, and in these experiments we placed each odour on one cage, and we had another cage as the last one that was our control cage that we placed all the odours on top of one cage. We did this test during 5 days of the peak of the reproduction of flies, and all the experiments that I explain you from now, they were performed under controlled lab condition, 28 degrees, 60% humidity, and 10 light day cycle. Based on the results, among the chemical odours from volatile component 1, 3, and 8, volatile component 1 was the best, and by increasing the number of components in the odor, we found that the efficiency of the odor to attract females reduces. Again, the DTI odor was the winner. That's our explanation is that our flies are completely adapted to this odor, so it's natural that they show a better response to this odor compared to the new ones that has been introduced to them. The lower graph shows the results of the control cage, and as you see, the control cage also shows the same pattern. DTI was the winner, and among the volatile components or chemical odors, only one component had the best results, and mix of 8 showed the lowest amount of X. Based on these results, we developed a new test where we compared volatile component 1, the winner of previous test, with a new component that I call it as component B, and mixture of these two. And in this case, we placed these odors on top, and we didn't use the DTI odor in this experiment. And we had, again, a control cage that we had a mix of all the odors. This test, again, performed during 5 days of the peak of the reproduction and under lab control condition. And we refreshed the odors every day. We also refreshed the odors in the previous test every day. We found that volatile component A, again, was the winner, and we were happy because it shows that we get a consistent results. And again, the mix of components had the lowest amount of X. That shows when we mix the chemical components together, it confuses the flies. And the control cage also showed the same pattern that convinced us to look more into the volatile component A, and it was our candidate for the further development, and we decided to optimize it based on the dosage and also the dispersing system in the next tests. As Queen explained, we developed 2 different dispersing systems. One is a simple one that is a water gel soaked in the odor, and you just need to make some small holes on the leads and place it on top of the egg collectors. And in another system, this container goes into a system that through some pumps, it distributes the odor on top of the egg collectors. Both systems works efficiently. We didn't see any difference between these two. The only difference is the workload. That the time that you need to load this system, that we call it a Smoctopus system, is much less because you need to just put few containers with a smell into the specific containers that you have here and run the machine. But in another case that you have individual ones, each one goes on top of the egg collector. But both works efficiently. So in the final test, we compared the volatile component A, our candidate, the right dosage that we found, and compared it with the DTI. But in this test, we diluted the DTI odor 10 times, and we didn't refresh the odors during the experiment. That was for 6 days, because we wanted to see the consistency of the odor during this period, that which one works better. And we collected the eggs every other day. Here you see the design of the experiments, and in this graph I show you the results. And as you see in this test, the volatile component A could hit the DTI. And when we looked at the amount of eggs we collected at each time point, you see here time point 1, 2, and 3. At the first and the second time points, DTI and volatile component A are going hand in hand. But in the last time points, the volatile component A could attract flies much better than DTI, which proves the consistency of this odor even without refreshing, and proves that if you use the right dosage of this component, it's a really good candidate for attracting flies, and it's a safe attractant. The next part is about developing egg collectors. In this regard, we looked at the color of egg collectors, shape, and material, and also we developed a new technique that enables us to collect eggs from the egg collectors without scrubbing and without damaging. Regarding the color, we tested egg collectors with different colors: black, blue, green, transparent, red, white, gray. And we did 2 tests to find that is there consistency between the results or not. In the first test, we had the highest amount of eggs from the blue and green egg collectors, as you see here in the upper graph. And in the lower graph, we have the results of the second test, and we collected the highest amount of eggs from the black, gray, and red. I have to mention that in these tests, I collected eggs for 24 hours, and we just used DTI attractant as the odour. So everything was stable and consistent except the color of the egg collectors. And as you see, we didn't find any correlation between the color of egg collectors and oviposition behavior of flies. Next factor was the shape and material. We developed different shapes that I show some of them here in this picture. And I can say among these shapes, the round one, the round egg collector, was the worst because the eggs stuck into the holes and suffocated. And the best ones were these egg collectors with the lamella, because it provides a good air ventilation. The light can penetrate easily through the lamellas, and it's safe, it's easy to maintain. You can wash it in the dishwasher, no matter what's the material. The green one is a soft plastic in this picture, and the black is a hard plastic. And it has a very low risk of contamination. So we decided in the next step, develop this shape of egg collector. And we looked at the distance between the lamellas to see that does it have influence on the amount of eggs we collect and the behavior of flies or not. We had egg collectors with distance between lamellas varies from 1.75 to 3.5. We did 2 tests again. And in both tests, we used DTI attractant. We collected eggs for 24 hours. And in both cases, we considered the egg collectors with 2.5 millimeter distance between lamellas as the control one. I have to mention that I did this test in 2 consecutive days. So as you see here, the results show that in the first test, the highest amount of eggs collected from egg collectors with 1.75, 2.5, and 2.75 millimeter distance. Please look at the blue columns. And the second test that is shown in red columns, the highest amount of eggs collected from the egg collectors with 3.5 millimeter distance between the lamella. So we again didn't see a specific correlation between the distance between lamellas and the oviposition behavior of flies, but it's worth to mention that we need to keep distance logical and we shouldn't suffocate the eggs. So something between 2 to 2.5 should be an optimal distance between the lamellas. Next feature of the egg collector was this coating one that our partner Jens came up with an idea to develop some materials to coat the egg collectors that enables us to wash eggs from the egg collector. So he developed 4 different coatings. The first and the second coatings were bio coatings. The third one was a chemical coating, and the fourth was a dissolvable fabric, as you see here in this picture. We did a test and we placed these egg collectors with the coated egg collectors on top of 2 cages, and we had 1 egg collector without any coating as the control one. The tests were performed, it was performed at for 24 hours, and again we used DTI attractant. In the first round, we found that the highest amount of eggs collected from the egg collector coated with coating number 2 in both cage 1 and cage 2, as you see here, this number 2 column here. And the lowest amount of eggs belonged to the egg collector that had been coated with chemical coating. But regarding to separation of eggs from the egg collectors, it was so easy and fast to collect the eggs or separate the eggs from egg collectors that had been coated with coating number 1 and 2 and coating number 3. But we also assessed the survival rate of eggs to see that the coating by itself and the washing eggs has influence on the survival rate of eggs or not. And we found that the survival rate of washed eggs from the egg collectors coated with a biocoating was 73%. That was approximately 15% lower than the unwashed eggs. So this process doesn't have any influence or doesn't have a significant influence on the survival rate of eggs if we use the biocoatings, but the chemical coating, we found that the survival rate of eggs was 60%. So these results plus the previous ones that we found convinced us to work more on the biocoating and develop some new questions in our mind. That could the biocoating by itself attract the flies without the presence of the DTI attractant? And is there any correlation between the flies' diet and their attraction towards the biocoatings? Because these two biocoatings that we have, the main difference is the presence or the amount of protein. Biocoating 1 has low amount of protein and biocoating 2 has high amount of protein. And since we had the least amount of eggs in the egg collectors coated with the chemical odor, we decided to look more into it and see that could we use this chemical odor as a repellent to prevent egg laying out of target. To find the answer of these questions, I developed a new experiment. And in this one, we use 2 different populations that we have in our lab. These populations have been selected on 2 different diets. One is the chicken feed and another one wheat bran for more than 20 generations. In the first 2 cages, we had the biocoating 1 and number 2. And in the 3rd and 4th cages, we had also the repellent 1. According to the results, surprisingly, we find that flies are attracted to the coating that is matched with their diet. So the chicken feed population was more attracted to coating number 2 that has high amount of protein, while the wheat bran population showed higher attraction towards the coating number 1. So there is a correlation between the flies' diet and the response to the coating, and also the coating can be used as an attractant by itself. And as you see, the coating number 3, we had the lowest amount of eggs. So this chemical coating has a potential to be used as a repellent, but we need to do further assessments to come and develop the right dosage of this chemical coating and also find the best way of using it or the implementation of this coating if we want to use it as a repellent. In conclusion, we developed several products during the FlyScent projects. One was a chemical attractant that is efficient, safe, and using the dispersing system that we have, we can say that it's consistent during the production of black soldier fly. We also developed an egg collector that is really easy to maintain. It can be reused, it's safe, and it provides an efficient way to collect eggs from the egg collectors without scrubbing and without damaging the eggs. Thank you so much for your attention and hope I didn't make you so confused.
Thank you so much, Anita. I don't think that's the case. We have some questions now, and I'll try to get an overview of it. And I can see as well we had some delays on some questions for Quinn's presentation. So hopefully you can answer like together on this. First of all, we have maybe the easiest to go to. How does Asian market smell?
It's actually like let's look at the sensory description of this one, but yeah, Asian market smell, you've got to go there to experience it. It's kind of like warm and smelly.
That makes everybody hungry.
Yeah, well, it depends on the market probably. Yeah. Yes.
Thank you. Thank you. I'll note that then. So in regards to did you test any volatiles in regards to fresh laid eggs?
This question from me or Queen?
I guess that's for you, Anita.
Yes, we did a test because we know that the eggs by itself has a potential to attract flies. We did a test and we used eggs, and at the same time we assessed the volatile components from the eggs. And the conclusion that helped us to find these volatile components, our candidates, our final candidate, was based on all these tests that we did. Yeah.
Awesome. Maybe a little bit related, but there's also a question in regards how much does these potential compounds be related to the food that the black soldier fly ate in their lifetime? And I guess that correlates pretty well to what you were talking about, the wheat bran and chicken feed generations.
That was about the coating. The coating, the first aim was not using it as an attractant. It was just surprisingly we found that, okay, it has also the potential to attract, and but it needs, if you want to make it more efficient, it needs the attractant by itself. We didn't find any correlation between the flies diet and our chemical attractant. We did the test on both populations that we had, and we got the same results. But the coating, yes, there is a correlation because of the presence of protein in the coating. There's a correlation between the flies diet and the response to these coatings, biocoatings.
Thank you. Next question is, can you measure the interactions between two or more compounds? I guess that's maybe on Quinn's side.
As in like chemically speaking, then you can mix as many as you like. But then we also did the test on on these compounds together with the flies, so we can totally mix the different compounds. And we actually did that. We have different matrices where we use like ranging from one compound to 8 different compounds. And but in the end, it seems that the fly lies more singular compounds instead of complex matrix. That's what we found out. But it's totally possible to mix them. Okay.
Yeah. So just also a shout out there that we can definitely test all your compounds you want to test in regards to the flies. So we as well have a question. Do you see any cumic effects? So any chemicals products that are necessary for the industrialization of this attractant on the BSF in long term? I'm not quite sure I understand the term cumic effect, but I guess, so do we see any correlation to if there's an absence of a specific chemical compound, is as I understand it?
Yeah, maybe we may come with another compound that works better than this one, but right now we compared it with one of the most efficient attractants that has been used by other producers as well, and it's heating that one based on consistency and also the possibility of or efficiency of collecting eggs. But everything is possible.
Yeah, but that's I think what the question relates to, if there's something that's absolutely necessary in order to collect the eggs. And I mean, the approach that we took looked at all of the positive correlations between the chemical compounds and the amount of eggs that we collected. So collecting data in that way will never reveal to us if there is one specific compound, because the different smells that we were using contained several of the same compound, almost always. So to figure that out, we would have to have bought a lot of single concentrated compounds and test them all. So we can't really answer that, but it's an interesting question.
So we have a question, or it's actually a question that several has been asking. So why are we Why haven't we tried lower size than 1.75 millimeters? We're talking about the egg distance.
It's just the separation, because one of the aims was coming with the distance that also enables us to wash the eggs from the egg collectors. So going less than 1.75, it was not possible to wash the eggs. And even in that one, it was really difficult to do that. So since it was a combination between the coating and the distance between the lamellas lower than that one, it was not good for our sake. But I can say that it has also influence on the amount of light that passes through the egg collectors, and some eggs get stuck between the lamellas if the distance is very narrow.
And there's one more thing: the distance between the lamella. The closer you go, the less volume for eggs you actually have, because the longer distance you can have between each of the lamellas gives you more volume, so you can collect more eggs on the same size egg collector.
Yeah.
Okay. We have a couple of questions, and believe me, they're not stupid because it's actually on purpose. So they're asking to what is the DTI attractant? And of course, that's something that we, for the moment, keep to ourselves a little bit, but that might be something that we can disclose on a later state. But Neda, you might want to put some words on.
Yeah. I agree with you.
Okay, good. So very politically moving past that. So we have another question, and I realized that the Q&A hasn't been working quite well. So I'll just take the last question there, and then we'll jump to the chat. So the question is, do you think it's necessary to remove the eggs from the egg collector, and if so, why?
To control the density for the juvenile, when you want to start a production, for the homogeneity of the juveniles. This is instead of, and you also get a fresh egg collectors. You don't need to wait till the eggs hatch and so on. It's just for these purposes.
And I can add from previous experience that it doesn't seem to decrease, at least the mortality or hatch rate, that leaving old eggs at least. But yeah, it's a measuring practical question, definitely. Good. So and then there's a question of whether we plan to test the attractant on a larger scale production. I guess that will be for the next presentation. So please hold that thought. And then how are you planning to create an appropriate FlyScent when there are so many different farmers, researchers, companies that are feeding their flies with different diets? Sorry, Neda, I guess that's for you.
As I mentioned, the chemical odor that we developed was not, it didn't show any correlation with the fly's diet. And the only thing was the coating. And when we mixed both together, we just improved the efficiency of collecting eggs. But yeah, the biocoating definitely is linked to the diet, but the chemical one that was the main finding as the attractant is not linked to the fly's diet at all.
But it is still a very valid question, because as you will hear from Jo after the break, without too much spoilers, it didn't work the same on the large scale as it did in the small scale in the lab at DTI. So it is a very valid question.
Yeah, and if I may add also, when you do, for example, experiments like this, that we see that most importantly for us is that now we know how to find these compounds. So whatever feed, whatever conditions you have, we can investigate how we figure out to have the correct attractants for you guys. So there's of course the application of it is not a one solution fits all, but now we have the method. I think that's an important point as well. I think the plan is now. I think that was all the questions, and please just write them in the chat now if there's anything I missed, because according to the plan we're actually just on time. That's excellent. So we'll have a short break, and I think you can just be in the lobby while we do the break, and then we will return in 10 minutes. I think it is.
Yeah.
10 minutes break.
10 minutes.
Yes. And feel free to ask questions in the break as well. We will we'll take care of all of it. Thank you for listening so far, and see you soon. Is it on?
Yep.
Amazing. That's good. Welcome back.
Just ask them that they hear you to be sure.
Can you hear me? Just make a
Thumbs up.
raised hand or anything. If this, yeah, that's amazing.
Perfect.
There was 85 people raising their hands. Simultaneously, awesome. So I'll move right to it. We had some questions in the chat, and we will wait with them till the open panel discussion we have just after Eurofear. So I'll introduce you, Yoav Politi, one of the 3 co-founders of FreezeM. He has a PhD in developmental genetics, working on Drosophila flies, early egg development in Weizmann Institute of Science. In FreezeM, Yoav is in charge of the breeding and operation of this, and he's doing, I must admit, some very innovative and interesting stuff, and it has been a really, really good part of this project. So the floor is yours.
Yeah, thanks. Do you hear me?
Yes.
Just fine.
Okay, thanks, Mats. Thanks everybody for joining and participating. Thanks all the guys in DTI for being such great partners, Jonas and Neda and Quinn. I'll present our work in this project, which was focused on several aspects, as you see. Mostly on the validations of the things that were developed under the DTI or by Jens and Dia, and some other interesting stuff. So I'll just share my screen. Let me know if you see it. Do you see it?
Yep.
Okay, so I'm happy to present our results as being 1 out of 3 partners in this project. So basically, I'm going to talk about 4 different topics. I'm going to show you some egg collection concepts that we developed here, and which might fit different schemes or potential egg collection methods. I'm going to present to you 2 different biological attractants that we were testing, and their ability to attract female flies to lay the eggs in our setting of the cages. And I'm going to talk about some validations that we did for the cassette coating that Neda showed, and also the chemical attractant that was showed very high potential in the experiments in DTI, and how they looked when we tested them in our hands. And as Jonas said before, also some troubleshootings that we needed to perform. So just to give you like a glimpse of how our cages look. Basically, our cages are a cylinder, and what we do is we place the attractant outside of the cage, similar to the one to the net cages in the DTI, but here it's the orientation is different. So you see on the left the attractant and the hose that is connected to, and you see like in the middle the how it looks from the inside of the cage, and how the egg collection looks like. On the right side, this is just an example of an adaptation of a cassette like the one used in the DTI and our scheme. Okay, so first the egg collections and the several versions that we were examining. So we got a special delivery from Denmark, their cassettes, and also they prepared for us a matching cassette, a silicone cassette, which will fit our hose. And as you can see on the right, they both work very fine. The control, meaning without any cassette, like just the hose as I showed you in the image before, and the cassette, which seem to be both attractive as it is, as a contact layer, and also its color. As you can see, 89% of the eggs were on the disc, which is a nice number. It's not close to 100, but it's fair enough to in this kind of cages to collect the majority of the eggs. We also tried two different other types of cassettes. These cassettes were of different concept, meaning that not only the eggs are laid on the cassette, we are able to disconnect the eggs from the cassette and then incubate it in the incubator when the eggs are separated from the cassette itself. So you can see different versions. These are the concepts. So one of them is a comb, and one of them is a card, which is perforated. Both of them worked nice, but they had some technical problems. First, they were tending to break, and they were made out of printed material, 3D printing, and they were less robust than other versions of cassettes that we were using. And another thing is that the release of the eggs from the cassette was sometimes harmful for the eggs. So in the end, the comb version on the left was the better option, but both of them need further development to make it efficient for industrial scale. The second thing that is very interesting that we were able to examine is and also to develop is two versions of biological attractant. You can see here three different types of egg laying in cages, in different cages. We score them, and this is what we use later on to evaluate how efficient and successful the attractant is. So you can see on the left, like, and also in the middle, less eggs in total. But also on the left side, you see that they are very dispersed and less on the middle, on the hose, which is the attractant releasing device. But on the right side, you can see how it looks like when the egg laying is you can say excellent performance, and the majority of the eggs are concentrated on the hose itself. So what I can tell you about the two attractants that we used, if we compare them, so the older version is let's say less comfortable to handle. It has a stronger scent, and it takes more time to prepare. And also before you prepare it, it has a shorter shelf life compared to the new version. So the new version was much more stable. The scent, the smell was mild and less and weaker than the other one. The preparation is relatively very easy, and they both last for the same time, meaning that once you place them on the cage, you can work with them between 1 to 2 weeks. And these are the results, which were very happy, and also eventually we stopped using the old version and start using the new version here in our cages in FreezeM. What you can see here is that we got from our cages, and I will show you in the next slide, some numbers about our cages, but we got about 42 grams per cage per cycle versus 38 grams per cage per cycle, which is still a very good number, but the other version, the new version, is much more successful, and that's why we moved to it. What you can see on the right is that the eggs were very much concentrated on the middle, on the hose itself, which means that the scent was, despite it was relatively mild, it was strong enough to attract and concentrate the egg laying there. If you compare, and if you want to get some numbers for you to take home, what are the number of flies that we use per cage? So it's roughly 4,000 flies per cage with a sex ratio of 50-50. If you try to extrapolate it and compare it maybe to your cage system, so if you take it over 1 cubic meter, so it's 151 grams of eggs per cubic meter per cycle. And our cycle is usually 7 days, so if you divide it per day, so it's 21 grams per day per cubic meter. So if you want to ask yourself if it's worth exploring more, so you can compare it with the numbers that you are familiar with, and I think it gives you like a good understanding of what we use here. The third part is the cassette coating. We were in contact with Jens and Neda and Jonas regarding the coating that they were testing in DTI. We received two versions of the coating, as Neda described before. Our setting of experiment was as following. We used 1D cassette, a square cassette, which held these type of attractants on it. So it was coated with the repellent. It was not coated at all, which is the 0, or it was coated, and on the bottom it was coated with the attractant. And we tried both attractants in different cages, attractant A and A1 and A2. And here are the results. You can see the cassette, and the orientation is the same. So what you see on the left top side is the repellent area, and on the bottom left side is the attractant area. And you can see in relative numbers the amount of eggs, or the percentage of eggs that were laid on the attractant area, which was 58%. On the repellent itself, we got 30%, almost none here on the non-coated area, and less than 10% on the outer, like outside the cassette itself. When we tried the second version of the attractant, we got somewhat better results. So the attractant was more efficient here. We saw less eggs on the repellent. Again, almost none on the non-coated area, but we saw more eggs on the outside, so not on the cassette itself. Now I have to say that we have to take into consideration that there are quite a lot of technical issues with these tests, and it took us some time to calibrate it, and I'm sure that the further calibration will improve the results and support the results that Neda showed with the possibility to attract the flies with these attractants and the coatings. The last part is the chemical attractant, which was described again in the previous talk. We used the same method, and we were guided by Quinn. So we had the same chemical compound. we diluted it and mixed it with these beads that were supposed to hold the scent. What we did is to try it in 3 different methods. We used our new version of the biological attractant as the control. We tried the beads only, and we tried a combination of the beads and the attractant of FreezeM. You can see here the results and how the egg collector hoses look like. On the left is the attractant itself, which we call the zoo. It's the chemical attractant by itself. In the middle, it's the control, and on the right side, it's the mixture. If you summarize the results and all the repeats, we saw that the biological attractant was better than the mixture and much better than the chemical attractant. Again, I can say here that as Quinn described, and also what we saw and we tried in our hands, the dispersion of the chemical attractant is very much important, and it needs further calibration in order to disperse it better. So what I'm saying is that maybe here what happened to us is that the dispersion was not good enough, and that's why we got like almost no eggs on the chemical attractant, despite that the good results that were shown in DTI. So if I may conclude, first of all, and as a bottom line that I can say for sure, is that the attractant performances are affecting dramatically the egg yield. And if the attractant works well, then you do get almost 100% of the potential eggs to be collected. The stable and high performance attractant can be derived from biological material. And this is what we've shown, at least in our case, in our cage system and on our colony, that the winner for us is the biological attractant that was developed during the project. And again, this is the one that we're using now, and we're very satisfied with the results. The calibration, as a conclusion, a painful conclusion, the calibration is mandatory, and it takes some time because one cage system and one colony scheme is different than the other. So despite the fact that there were very much promising results in one place, it doesn't make, or it doesn't ensure us that they will get the same results immediately on the other system. And another thing, which again is that we need to take into consideration that after we collected the eggs, we also need to take to consider how we incubate the eggs, if it is with the cassette, without the cassette, and how we collect the neonates afterwards in order to proceed the process and see the neonates properly. Okay, so that's it for me. Thank you all for listening, and yeah, if there are any questions.
There are many.
Thank you so much, Joe. There are definitely some questions. There is as well some from the, as I said, the last part. So I think we'll keep your questions to you now, and then we'll take the rest afterwards. There's a question that flows around. A couple have asked. So how much cubic meters is your cage, and is it 4,000 flies per cubic meter?
Okay, so our cages, as you saw, are the cylinder cages. Those are 0.28 cubic meter. So it's 1 meter long and 60 centimeter in diameter. So you can make the calculation. So it's less than a third of a cubic meter in diameter, in volume. The 4,000 flies is per these cages. So if you want to make the extrapolation for bigger cages of 1 cubic meter, you can do so.
Okay. Next question is and down to how do you measure 50-50% sex ratio?
Yeah, so these are according to our experience and tests that we ran internally. We know that our let's say the timing of the emergence from the pupa is so that we get 50-50, around 50-50. Yeah, it might be 45-55 or vice versa, but we are able to control the sex ratio very accurately, relatively very accurately, and we do, like, before we close the cage, we are able to monitor also like the sex ratio and see that indeed, like when you sample the flies, you get like around 50-50.
Okay, thank you. So, and there's I think that's as well a practical question to your fly cages. So, what is attracting the flies to oviposit around the perimeters of the hose and not in the center of your setup?
I think also Neda suggested that it also related to the orientation of the light. But I guess also the scent, the smell is strong enough to attract them to this area, and they do lack the contact. So eventually, as you saw, it's a net, and you get like the surrounding of the hose. So they prefer like to lay the eggs on the surroundings. Sometimes it's more on the inside, sometimes it's both on the inside and the outside, but it's good enough for the flies, I guess, that to feel the scent and also lay the eggs that area. And we do see that these concentrations very it's very clear that they do prefer this area on any other place in the cage or outside the cage.
Okay, yeah. Okay, makes sense. So I think we have 2 questions more related to you solely, and then afterwards we'll go into like more an open discussion. So what is the longevity of the flies in the cage?
Well, our method with these cages is batch wise. So usually our cages last for 1 week. Not because after 1 week they stop laying eggs, but in terms of efficiency, we prefer to just discard the flies and reopen the cage with a new batch. So you can take it for more than 7 days, but if you try to make the maximum eggs per day, then it doesn't make sense to last it for day 8, 9, and 10.
Okay, thank you. Yeah, I actually think that the last question you already answered that, but I'll give it a go again. So how do you control the sex ratio when the males are known to emerge earlier, whereas female emerge later?
Yeah, so again, these are small techniques that we developed here in house. We maintain the flies in different, like every developmental stage is in different room, in different conditions. So we are able, when there is this transition between the pupa and the adults, we are able to control the sex ratio and then insert it to the cage on the right timing. So you'll have like overall a 50-50 sex ratio in the end.
Good. Thank you so much, Yoav. And there is a question I missed. So how do you control the fly density and as well the sex ratio across the cages?
And the difference that they have seen is the continuation of this question, that the difference that he has seen is due to the density or more females or everything is under control.
Yeah. So again, we as I said, we work in like batch. It's not a continuous cage. So we place the emerging flies or the pupa and the transition in the cage, and we take it out. So we do have like a narrow window of emergence of adults, which we control, and that enables us to keep these 7 days of cage opening and also the sex ratio. So of course, if you place pupa, like when the batch start to emerge, then you will get, as some of you said, more males and then half and half and then more females. But the synchronization doesn't make sense in this, let's say, large scale production. So our technique is to hold it for a short period of time and then take the pupa out, the remaining pupa out, and then you do get this 50-50 sex ratio.
Yes, makes sense. I think if I haven't missed anything else, and specifically to you, Jorf, I will say thank you so much for the presentation. And we will move into the, let's say, the more panel-wise discussion where probably all of us can pitch in. We have first a question that was left in the previous one, which is, did you test the DTI attractant with different genetic strains? And if yes, how did the results vary? I think, Nida, that's one for you.
Yeah. I would like to start with this study that mentions all the strains that we have in Europe. They all arise from one population from America. So all the populations that we have, even though they have been adapted to different diets, but they arise from one specific, or they have a specific genetic background. Based on this study. So we haven't tested on different genetic strains, but it has been tested on populations adapted on different diets.
Thank you so much, Nida. Very precise, good answer. So next question is about the egg collection. How was the lighting around the egg collectors? Literature talks about laying eggs in dark places.
The light condition we have in our lab is from top, but as far as I know, black soldier fly prefers to see the light, and after that light also plays a role as an attractant, and they fly towards the light and lay egg in a light place. So but it depends on the density of the light and the looks of the light and so on. But we have the light, LED light, if I say correctly, and it's from the top.
Jorf, would you like to add to that point as well?
You are mute, Jorf.
Yeah, sorry. So first, I think one of the attractants that for the first question that we tried here is similar to the DTI attractant. So you can say also that we tried it in various colonies, because our colony is not the same as the one in DTI. And referring to the light, so yeah, the light, well, yeah, it's mostly for the mating, but it has also influence on the orientation, or you really see it, like the pattern of the egg laying is really related to the source of light. So it is related, and it's something, I think it wasn't on the scope of this project, but it's something to further quest.
Thank you for that point. So next question is, I think it's more like a practical to the results you presented, Anita, is, are there any eggs that were stuck in the cage nest while the egg collectors were removed, which result in some loss of eggs?
There are some individuals, but it depends on the size of net that you use. In our case, it's just very few individuals. We get most of the eggs on the egg collector.
Good. And then we need a very practical answer to this question. So would you share how you wash eggs from the biocoating or chemical coating, and what is the process of that?
I did 2 different processes, and the one was soaking the egg collector in water, and another one just washing right away under the tap water. And the tap water was faster, efficient, and just by keeping under water for a few seconds, all the eggs just fall down.
Very interesting.
And the survival rate I showed you is from that method of washing.
And then again, maybe a technical, I think Jonas as well can answer on this. This is why did we pick silicone?
Yeah. So the reason that we picked silicone is because we used it before. I started using the silicone way back when I started here working with the mealworms. The mealworms also glue their eggs, and we found that the silicone did not attach very well to the eggs, so we could collect the eggs from them. That's why we kept using the silicone to make the egg collectors for the black shoulder flies. Also because it's very easy, because we can 3D print a cast, put the silicone in, and then we can make several egg collectors every day. So it's because we did it before, easy, efficient, and the silicone is food grade, which was also something that we had to consider at a point.
Yes. And then we have another question in regards to, does the biological attractant in this case is still the result of fermentation or synthetic and presented on beads? I missed this. Was the question?
It's from Jov.
Yeah, so your biological attractant, Jov.
Yes.
Yeah, I'm not sure I understand, like, what is the question?
So is the biological attractant you did, is that still a result of fermentation, or is it synthetic, or is it, let's say, picked up from the ground? So
yeah, so my guess is maybe it's not like deliberately, but it's a case of decaying and like fermented in the end like material. So it is biological and not like a single molecule. Which was more of the chemical attractant. So it's like let's say a whole, or like various processes that happens on this biological material that makes it so attractive for the flies in a way that it mimics the process that happens in nature, you know, that these flies are attracted to composters and decaying matter. But in the end, we were able to recapitulate it and like mimic it in a way that you can prepare it in advance and store it, and you get the same results as just taking like rotting stuff from outside. So I guess there is like the fermentation there and the fungi and like bacteria that are there, but you know, I cannot I don't know like what exactly happens there, but it works, except for the fact that I know what is the material and how to prepare it.
Okay, yeah, that is as well important to know that as well. So I have a combination of 2 questions. I think I'll answer or ask Neda as well afterwards. So that's how many eggs are you getting per female, and what do you think the maximum size of a cage would be for this attractant to work? So it's kind of like 2 questions in one. Do you want them repeated?
For me?
Yeah, for you in the beginning.
So again, I didn't make the calculation. If we're talking about 4,000 flies per cage, 50-50, so 2,000 females. Make the fast mathematics. If you get 45, today we get even like more than 45 grams per cage per cycle. So you can make like this calculation of how many eggs you get in the end. The size is a good question. Yeah, of course, and this is also something that we try to address in this project, is the dispersion and how strong is the scent that will attract the flies from a larger distance. And this is something that we're further examining. We have these type of cages, as I showed and described, but it needs to be further examined in bigger or smaller cages. Like it's not that if you go to smaller cages, it will perform the same, because it might be too strong for these cages. So it works on in these settings for the 1 meter long cage, but it needs to be recalibrated for bigger cages, I'm sure of that.
Thank you.
If I can add here, I think the size is not, unless you go ridiculously large, it's not a problem. It's just, in my opinion, it's more of a question of how you distribute the smell to make sure that you have a gradient of concentration that the fly can fly towards, so that it can follow follow the strength of the signal and find the egg collector where the scent is strongest, because the molecules are not going to degrade over such short distances. So I think it's much a question of airflow and how you control having low concentration in one end and high concentration around the egg collector.
And the time that they provided for flies, they should provide at the right time before they start to lay egg. True. In other places.
Yeah.
I just, maybe I didn't say it just as a comment to my presentation. The biological attractant was not like replaced during or not renewed during the lifetime of the cage. So it was one time, and of course the timing when you place it after you open the cage, so it has an effect, because you might get like a nucleation of the egg laying in other places, and then you lose the entire rhythm of the cage. And yeah, I agree with you, honest. Like unless you go really big, like a few times bigger, then you just need this gradient that the flies will feel and like go towards the smell.
Thank you. Okay, so the next question is, did you investigate the effect of the collector design on the neonate hatching rate, if any, due to possible factors like ventilation, condensation of the design? So I think you already touched a little bit upon it, but please feel free to add.
This is about the coating one, the coated egg collector?
No, the design of the egg collector.
Yeah,
the
design of yes.
I didn't look at the survival rate, but we didn't see anything because they all were. But I didn't look at, to be honest, I didn't look at the survival rate. But we could see that from the different sizes, they could hatch. But the issue was that we collected because we had the coating. It was the size plus the coating. We washed from them. So I cannot say that the eggs were on the egg collectors during the hatching period, maybe we could see the different results. No, I didn't check that one.
Okay, no worries. So in regards to the next question, it's something that I have definitely never heard of. So maybe someone can answer that. I heard a technique of putting talcum powder in the eggies. Then you shake the eggies and the eggs will fall. Have anyone tried this? It's maybe like maybe you maybe nodded.
Like talc powder?
Yeah.
No.
I no.
I'm very interested to do it now.
Yeah.
That must be the next big thing. So we have a question as well where we have a natural behavior of black soldier flies to lay where others have laid. Definitely agree. And if there's any research to understand if these eggs themselves are an attractant scent profile, and this actually leads quite well into probably something I'll mention later. But some of the things we do at DTI right now is focusing on exactly that approach. We want to see whether we can further develop on this kind of scent attractant, and especially looking at the eggs, because we've seen it not just with black soldier fly, but for other insects that this is an excellent attractant. So that's definitely in the scope for our next big project.
And we did a test in this regard that we used a fresh egg as an attractant and compared it with a DTI, and they went hand in hand. It was as efficient as the attractant. And in the first trial, because I always say that about the chemical attractants, the adaptation is also plays role. In the first generation, maybe they don't show response, but in the second trial, the third trial, they, as you see, even in my results, in the third trial, we got the best results. But in response to egg, they show immediate response. So it's a really good natural attractant.
So and that's an extension to our question. Now the chat is moving a lot. So did you take a visual attractiveness into account as an effect on the collector design? I guess that comes down to
the color. Yes.
Color, yeah.
Not in other.
Even I did more than 2 trials. I didn't show the results. We didn't give up. But at the end, we came up with this conclusion that no, there is no correlation between the egg collector, the color of the egg collector, and the oviposition behavior of flies. So maybe about the color, they are colorblind and cannot see, but yeah.
I can say from only, this is not in DTI regards, but from our own, we tried as well a lot of different metals and wood substrates in this kind, and I think the only thing we saw had a negative effect, I think, was very, I would say not bright, but stainless steel wise, it seemed to not be working as well as others, but in that result, I don't think in the visual effects I've been seeing other than that. But I guess that's due to the reflection of light. Maybe that's confusing them in some way. I need to check on the time, but I think we have time for a couple of questions more. And this is toward the size of the cage. What is your opinion on how measured egg yield? Eggs per square meters per day or eggs per cubic meters per day? So that is. I need to elaborate, sorry, it's eggs per square meters, so sitting area for flies per day.
Yeah, so eventually, I guess, when you want to be as efficient as possible, you want to take into consideration the footage or the size of your room. So if you can walk vertically, I don't know, 6 meter long height, sorry, so you're more efficient. We tried, in order to make it universal, to express it in cubic meter, but of course there is also like you need to take into consideration like the area that you are holding. So it depends on like a point on your point of view. There is no like a right answer.
I would say we actually tried in from my firm of production. We tried doing it at eggs per square meters of sitting area, because we saw this ratio meant a lot more than the cubic meter. But again, it comes down to the diameter. So are you using cylinders? That is not as important probably as it is if you're using cubic squares or how you do it. So definitely there's something to come with there. But I think the sitting areas, at least for my opinion, is an important point of the efficiency of your density, definitely. And there's a question more. Is there any effect on neonates hatch rate from different attractant, biological attractant or chemical attractant? I think Neda, you could probably answer that.
No, about the hatching rate, there is no difference, but I would like to mention that when we use the chemical attractant, flies laid egg in a shorter window. It might be due to the influence on the fertility in a shorter period or the lifespan of the flies. So we were able to collect eggs in a shorter period compared to the biological attractant.
Thank you so much. And I think we are almost at the time where we need to be closing down. I'd just like to have some final remarks. First of all, of course, we at the DTI has found this project very, very interesting. And this, of course, have treated our interest to look further into this, because I think there's a great potential in looking in more in depth in whether can we find more things that the flies more generally like, or specifically for feed, or is there any repellent we can develop further? And that's definitely something we'll look at DTI. And I'll also use the time just to quickly say that we as well are working on a big scope on doing fermentation as well, so pre-treatment of different kind of feeds. So not related to the FlyScent, but of course I'll take the time to share that as well. And as well, we're working on the non-food part of insects, which I would love for you guys. If you had some ideas or thoughts about that, just contact me. It's something that we work on as well. And I think that will just be my short introduction to what we'll be doing the next couple of years, and hopefully also having webinars on that. And on that note, I think I will be closing the webinar for now, unless there's anyone in the panel or Europe that wants to conclude something. But at least, as we say, thank you for peace and order, and not too much shouting on the line. So amazing, and thank you to all the presenters. It has been very interesting. And don't hesitate to contact any of at least for our side, DTI, if you have further questions or something that you need to have elaborated. And ask us if you want to have the video is recorded. Just write us an email and we can figure it out. Thank you so much for your time, and that will be it.
Thank you.
Thank you.
Thanks a lot.